understand it.
All right, so I think everybody got it. So in the first step, we generate hydride by deprotonating either the oxygen or the carbon, choose whichever you want. I think it's easiest to deprotonate the oxygen, but whatever, right? So the general idea here is that we have a general base. We deprotonate either the C or the O. Again, I don't care which. Move the electrons over, make your carbonyl, and release hydride. That hydride attacks NAD, move electrons through NAD down to electron sink to give us NADH. So the redox step happens first. In the next step, we have to decarboxylate this intermediate oxalosuccinate. Okay, so this happens in this manner, right? So we get our decarboxylation. Let's redraw this. We have this and we have this. So the electrons flow down. The electrons in this bond move up to make a double bond, and we pick up a proton. Okay, that gives us this intermediate where we have an enol, and then we just kind of reverse the alcohol formation. So we have a base that picks up that proton, electrons move back out here, we have a general acid, and we pick up a proton from the general acid. Okay, so reasonably straightforward chemistry. The only tricky part here is to understand that it goes in the direction first oxidation, then decarboxylation. Right, that's different than the pyruvate dehydrogenase complex, which first we saw decarboxylation and then flow of electrons to NAD+. Questions about it? All right. We have now made alpha ketoglutarate. Okay, alpha ketoglutarate is our second alpha keto compound, and its fate is oxidative decarboxylation to make succinyl-CoA. This is the reaction I said that looks identical to pyruvate dehydrogenase. Step 1, decarboxylate. This carbon gets linked up to TPP. We then transfer this entire carbon chain onto lipoic acid. We then transfer the entire carbon chain onto coenzyme A. That gives us our succinyl-CoA product and our CO2 product, but we haven't generated NADH yet. Instead, right now, those electrons are trapped in lipoic acid. Lipoic acid passes their electrons onto FADH2. FADH2 passes electrons to NADH. Okay, I realize I just talked through that and you had nothing to look at, but the process, the flow of electrons, the flow of molecules is absolutely identical to the pyruvate dehydrogenase complex. Okay, so understanding that mechanism lets you understand this, because the only thing different is what hangs off of the alpha carbon. Nothing else changes. Okay. Succinyl-CoA synthetase. This is a cool one. Okay. So the idea here is that we are harnessing the energy in this thioester bond. We are hydrolyzing it. And we're using that energy to make ATP from inorganic phosphate and ADP. Okay. But remember, a big theme I've tried to stress this entire semester is that we can't just magically transfer energy, right? We can't just hydrolyze this bond and then give that energy to ADP and phosphate. We have to transfer that energy through covalent bonds. Okay, so before I move on, somebody proposed something that might happen here. How can we hydrolyze this and trap that energy in the form of a covalent bond? I believe Kenny's eyes just said it all. He goes, think back to GAPDH. Way back in GAPDH, we had a thioester. Anybody remember what happens next? Gabby. she's like, shit, no. Anybody? Okay, so So think back to gap, think back to gap DH, right? With gap DH, this is where we create our first high energy phosphate compound. We create carboxylic acid that has a phosphate on it. We create a mixed anhydride, and that's a high energy phosphate compound. So with gap DH, this thioester bond is replaced by a phosphate. Is there any way that might happen here? Somebody say yes. Thanks. Yeah, so this inorganic phosphate is a nucleophile, attacks this carbonyl, the thioester bond is broken. Breaking that thioester bond provides enough energy to phosphorylate this carbon. We have seen that before. We saw that with GAPDH. That's how we make 1,3-BPG, which then has enough energy to make ATP, right? This step happens within the active site of GAPDH, okay? So inorganic phosphate attacks, we break the thioester bond, we make an intermediate where we have this succinyl phosphate. And then that can ultimately be used to make ATP. But there is actually an intermediate step. Okay, so the chemistry here, we have our inorganic phosphate serving the nucleophile, attacking and breaking the thioester bond. That gives us this succinyl phosphate intermediate. Okay, the unexpected next step is actually transferring the phosphate off of succinate onto this histidine in the active site. So we make a phosphohistidine intermediate. Somebody remind me what enzyme used a phosphohistidine in the active site? The one I showed it, not so. This is a glycolysis enzyme. The one that converts 3-phosphoglycerate to 2-phosphoglycerate? Mutase, yeah, phosphoglucomutase. So, sure you were. So so remember, phosphoglucomutase uses this phosphohistidine as a source of phosphate to put a phosphate onto 3-phosphoglycerate, and then we put the phosphate back onto the histidine when we generate 2-phosphoglycerate. Okay, go back and review that. You've seen it before. It happens. So here, this histidine reaches out, removes the phosphate from our succinyl phosphate, generating our product, succinate. The energy is trapped in this NP bond. So, and then ATP or ADP comes in, removes the phosphate off of histidine, we get ATP. Okay, so the idea here is that hydrolysis of this thioester bond, that energy is translated through 2 different covalent bonds, ultimately allowing ATP to be generated from ADP and inorganic phosphate. Okay, if there's one theme that you take from the chemistry in this class, it's that energy transfers through bonds. Okay, this hydrolysis energy is transferred through this mixed anhydride bond and then through this phosphohistidine bond. And that's really all the mechanisms we're going to talk about. Well, until next week, Kenny. So the rest of the chemistry that happens in the TCA cycle is pretty straightforward, right? It is more simple pieces than what we've observed so far. Okay. So let's think about the overall process. The overall process, we bring 2 carbons in the form of acetyl-CoA. Those 2 carbons end up becoming carbon dioxide. So acetyl-CoA is a reactant, 2 carbon dioxides are products. The coenzyme A gets regenerated so it can be reused in the cell. In addition, the carbons in this acetic acid are oxidized, generating a total of 8 electrons. Those 8 electrons are stored on 3 units of NADH and 1 unit of FADH2. Okay? These are important molecules that get reduced because they're going to be the source of electrons for the electron transport chain. In addition, we harness the energy of thioester bond hydrolysis to make one equivalent of either ATP or GTP, depending upon what organism. This image shows GTP, but it could just as easily be ATP if it's a different organism. Okay? So remember though, this whole process, right? The conversion of pyruvate to acetyl-CoA going through the TCA cycle happens twice per glucose, right? Because glycolysis splits the 6-carbon chain into 2 equivalent 3-carbon chains, generating 2 equivalent pyruvates, generating 2 acetyl-CoAs. So all of this actually happens twice when we're thinking about starting from glucose. Okay? The last big thing to think about here is the energetics of this process and regulation of this process. Okay? So energetics, most of these steps are either at equilibrium or spontaneous under standard biological conditions. The 2 steps to really focus on here are the aconitase, isomerizing citrate to isocitrate, and then the last step, converting malate to oxaloacetate. Note here that both of these are endergonic, especially the malate dehydrogenase step. It's highly endergonic. Okay? So the way that we can push non-spontaneous reactions forward is by impacting the concentrations of reactants versus products, right? We know, remember, go back to delta G equals delta G naught plus lnq, RT lnq, right? So impacting concentrations of reactants and products can help us overcome otherwise non-spontaneous reactions. And that's what happens in the TCA cycle, is for the aconitase step, you know, it's reasonably close to equilibrium. It's not super far away, but nonetheless, this is non-spontaneous under standard conditions. But note that the previous step is highly spontaneous, and the subsequent steps are also highly spontaneous. So the idea here is that the previous step is spontaneous enough that we start building up lots of products, right? Or for the aconitase reaction, those are reactants. Because the previous step is spontaneous, we have a buildup of the reactants of the aconitase step. And because the next steps are very, very spontaneous, we have a depletion of the products. So we have a buildup of reactants, a depletion of products, which is going to push this non-spontaneous reaction forward. The same thing happens down here with the reaction 8, with malate dehydrogenase. And this is actually the really important one. Oxaloacetate, the concentration of oxaloacetate in the mitochondria is absolutely critical. Okay, it has to be maintained really, really well. It has to be maintained really low. So that at any given time, the concentration of oxaloacetate in the mitochondria is exceptionally low, because if it's not, then this reaction cannot happen because it's so endergonic. So the way this reaction is the way it actually happens is because the subsequent step, conversion of oxaloacetate into citrate, is very spontaneous. So we actively deplete the concentration of oxaloacetate to drive this reaction forward. In addition, remember that whenever NADH gets produced, it's pretty much immediately consumed in the mitochondria, right? Those electrons are pretty much immediately given to the electron transport chain. So not just depletion of oxaloacetate, but also depletion of NADH. And the final thing is the proton there, right? So we are in the matrix of the mitochondria. And if you don't know already, you'll know next week, right? The way that the mitochondrial electron transport chain works is it pumps protons out of the matrix into the inner membrane space, right? That's the proton gradient we're talking about. So in addition, we deplete the proton concentration. So every one of the products of this reaction get depleted, which lets this reaction be driven forward. Make sense? All right. Regulation. Everybody's favorite thing, right? Right? Thank you. So control of metabolic flux makes sense, right? With glycolysis and gluconeogenesis, we talked about it from 2 perspectives. One perspective is controlling flux through these pathways based on the cellular energy charge, as indicated by ATP, ADP, and AMP levels. Okay? The other thing that's important is hormonal signaling, right? The communication with the rest of the organism. Okay? And We know how all that works with glycolysis and gluconeogenesis. Similar things happen with the TCA cycle, and one of the most important regulatory points is the conversion of pyruvate to acetyl-CoA. So the activity of the pyruvate dehydrogenase complex. It's strongly inhibited by high energy charge of the cell, right? So a high energy charge, meaning a buildup of ATP, means the cell has plenty of energy. Let's not waste resources to make more. So a high ATP concentration is highly inhibitory of the PDH complex. In addition, a buildup of NADH signals plenty of energy in the cell. So that inhibits the PDH complex. And there's direct product inhibition. A buildup of acetyl-CoA also inhibits it. So lots of downstream signals will inhibit the pyruvate dehydrogenase complex. And if this complex is inactive, we're not converting pyruvate to acetyl-CoA, so there's no way to enter into the TCA cycle. Okay? The opposite is true at a low cellular energy charge. Under these conditions, we have a depletion of ATP. We start having a buildup of ADP and AMP, and ADP is activating. This is kind of interesting to me, right? Because we talked about with glycolysis and gluconeogenesis, it's all about AMP levels, and we talked about why that is. Because as ATP gets really, really low, ADP gets converted to AMP and ATP, right? So here's a situation where it's not AMP, but it's ADP. So this kind of gives this really cool graded response. In addition, the other thing to remember is that this reaction happens in the mitochondria, not in the matrix, or not in the cytoplasm. So ADP is highly present in the mitochondria, right? Because ADP gets phosphorylated in the mitochondria. So a buildup of ADP really means low energy in the mitochondria. Make sense? Okay. So the way that this enzyme responds to cellular energy charge makes a lot of sense. Okay? In addition, this enzyme responds to hormonal signals. Okay? The activity of the pyruvate dehydrogenase complex is dependent on phosphorylation. If we phosphorylate the pyruvate dehydrogenase complex, we inactivate it. Okay? And the really great thing is the way this connects to glucagon and insulin is not inverse of what makes sense. This one is directly makes sense. It's a kinase that phosphorylates PDH to make it inactive. It's a kinase. Glucagon activates a kinase. Glucagon is secreted in response to low blood sugar. When there's low blood sugar, we want catabolic pathways to be turned off. We want gluconeogenesis to be activated. So when glucagon is actively secreted and sensed, we have an active kinase that phosphorylates the PDH complex, inactivating it. This prevents glucose from being funneled into the TCA cycle. This prevents pyruvate from being converted to acetyl-CoA. Okay? That basically pushes things back through gluconeogenesis. The opposite's true when insulin's secreted. When insulin's secreted, there's plenty of sugar in the blood, right? We don't need to mobilize sugar in the liver to pump into the blood. Instead, we want to go ahead and activate the PDH complex so that the glucose that's coming into the cell can be used for energy. So remember, insulin activates protein phosphatase 1, so it activates phosphatases. This phosphatase hydrolyzes the phosphate off of PDH, activating it. So the TCA cycle gets activated. Yeah. Yeah. Exactly, right? So inactive PDH means there's no entry point into the TCA cycle. So any pyruvate has to be diverted somewhere else. And what we've talked about so far is that is going to be gluconeogenesis. So pyruvate gets diverted back through gluconeogenesis to make glucose. This is especially important in the liver because the responsibility of the liver is to mobilize glucose and put it into the blood to fuel other tissues when there's a low blood sugar level. So when glucagon is being secreted, it means there's low blood sugar. This makes sure that all pyruvate can be diverted back through gluconeogenesis. All right, there's two other regulatory points directly in the TCA cycle, but they don't respond to hormones, okay? These are just feedback regulation. So high ATP and NADH concentrations inhibit isocitrate dehydrogenase and inhibit alpha-ketoglutarate dehydrogenase. This makes sense. A buildup of cellular energy says TCA cycle doesn't have to be active. And a buildup of ADP is activating. The very last thing, and I'm going to finish a few minutes early. First time ever. So this is the idea of anaplerotic reactions. Anybody know what anaplerosis means? I never knew until I studied this either. So anaplerosis basically, it means replenishing. Okay? So it is really important for a cell to be able to maintain a steady concentration of all of these TCA cycle intermediates, possibly more important than any other metabolites in all of the cell. Okay? So that would be easy if there was no other fate of these molecules, but there is, right? This is such a central pathway that lots of other pathways branch off of it. And this just points out some of them. Citrate's important for fatty acid and steroid synthesis. Alpha-ketoglutarate is really important for the biosynthesis of some nucleic acids, our purines, and lots of amino acids. Succinyl-CoA is important for the biosynthesis of porphyrins and hemes. Oxaloacetate, amino acids, and our pyrimidines, right? So the idea here is that to be able to make fatty acids, to be able to make proteins, to be able to make heme groups, to be able to make nucleic acids, we have to siphon off these intermediates.